Hello friends, today we are going to study mutations. A change in the sequence of bases in DNA or RNA is called as mutation. Everyone has mutations. In fact, most people have dozens or even hundreds of mutations in their DNA. Mutations are essential for evolution to occur. They are ultimate source of all new genetic material, new alleles in species. Although most mutations have no effect on the organism in which they occur, some mutations are beneficial. Even harmful mutations rarely cause drastic changes in organism. So, mutation, that means change in the nucleotide sequence of DNA. It may occur in somatic cell, but it doesn't transmit it into the next generation. It may also occur in gametes, that is eggs or sperms, but it is transmitted into the next generation. There are two classes of mutation: spontaneous mutations and induced mutation. Some mutation seems to happen spontaneously without any environmental influence. Those are called as spontaneous mutations. These are occur when mistakes are made during DNA replication or transcription, while other mutations are caused by environmental factors. Those are called as induced mutation. During replication or by incorporation of incorrect nucleotide, spontaneous mutations occur. It is the change in DNA sequence or nucleotide sequence during replication. Induced mutations are due to some environmental factors. For example, UV light, X-rays, gamma rays, etc. Now we will be going to study different types of mutations. Mutations are of two types. One is chromosome mutation and another is gene mutation. So whenever there is a change in structure of a chromosome, that is loss or gain, then it is called as a chromosome mutation. Chromosome mutations are of four types: deletion, duplication, translocation, and inversion. First is a deletion. So here chromosome breaks at two positions in the chromosome, and that portion is deleted from the chromosome. So due to the breakage, a piece of chromosome is lost. So there is a loss of genes, that is called as a deletion. Deletion is always lethal because there are loss of some important genes. The second type is a duplication. Whenever a chromosomal segment breaks and gets duplicated and it is incorporated in a chromosome, it is called as a duplication. So duplication, there is no loss of genes but duplication of genes. So duplication is not much harmful as deletion. Third type is inversion. Here also chromosome segment breaks, then the segment flips around backward and again reattaches. So there is no loss of genes, only the sequence or order of genes in the chromosome have been changed. The fourth type is a translocation. It is the transfer of genes between two chromosomes. So it involves two chromosomes which are not homologous. There is exchange of chromosomal segments. Here in this diagram it is shown that there are two chromosomes. At one chromosome, the certain part is deleted, and the second part, in the second chromosome also certain part is get cut, and there is the exchange of these parts between these two chromosomes. So after translocation, the sequence of genes on these two chromosomes has become changed. The another type, that is the gene mutations. So whenever the change occurs in a nucleotide sequence, that may be single or in a genetic code, then it is called as a gene mutation. Gene mutations occurs due to copying errors, chemicals, or some viruses also. Gene mutation is also called as a point mutation because there is change in a single nucleotide. It includes deletion, insertion, and substitution of one nucleotide in a gene. So there are four types: insertions, deletions, substitutions, and frame shifts. In insertion, by an accident, an extra DNA base or bases are added to the DNA. So there is a production of wrong amino acid. It may be possible for termination of amino acid chain, that is protein chain, or there is a change in amino acid which may be responsible for producing non-functional proteins. Insertion can occur anywhere in size from one base pair incorrectly inserted into DNA sequence to a section of one chromosome inserted into another. So the examples are Huntington disease and fragile X gene, where trinucleotide repeats are inserted into the DNA sequence. The second point mutation is a deletion. Whenever an error occurs in a single nucleotide and then get deleted, then it is called as a deletion type of mutation. So here in this diagram it is shown that initially the codons are like ATG codes for methionine, AGT codes for serine, CGT codes for arginine, ATA codes for isoleucine, and TAA is a short codon. But after point deletion, that is in the genetic codon AGT, T is get deleted. Hence, the sequence of genetic codon becomes like this: ATG, AGC, GTA, TAT, A. So ATG codes for methionine, AGC which is changed. So again it is code for serine. GTA codes for valine. So there is a change in one amino acid, that is at third position. Initially there is arginine which is changed to valine after the deletion of T, and the further amino acids synthesized is tyrosine. So there is change in the function of amino acids due to change or deletion in a single nucleotide. Deletion can also range from single nucleotide to entire section of chromosome. Deletion also results into frame shift that changes the reading frame of subsequent codons and therefore alters the amino acid sequence which follows the mutation. Deletions are always more harmful. The diseases due to deletions are cystic fibrosis, Turner syndrome, and Williams syndrome. The next point, mutation, is substitution. Sickle cell disease is a very good example of substitution. It is due to the result of one nucleotide substitution, and it occurs in the hemoglobin genes. In the normal hemoglobin, in the beta chain, there is a amino acid glutamic acid at the sixth position. But due to mutation or substitution, the glutamic acid is replaced by valine, and due to that replacement, the disease occurs, that is sickle cell disease. There are different types of substitution mutation. Transition. If a purine changes to purine or pyrimidine changes to pyrimidine, that is A changes to G or G to A, or C changes to T or T to A, then it is called as a transition type of mutation. Whenever purine nucleotides changes to pyrimidine or vice versa, then it is called as a transversion type of mutation. A changes to T or T changes to A, or G changes to C or C changes to T, G, then it is called as a transversion. Substitution mutation is also of three types, that is a silent mutation, missense mutation, and nonsense mutation. Whenever the change in the codon but there is no change in the protein, then that type of mutation is called as silent mutation. there is a change in codon and it codes for a wrong protein, then that is called as a missense type of mutation. And whenever due to the change in codon, there is a termination of protein chain, that is further protein chain synthesis stops, then it is called as a nonsense type of substitution mutation. See in this diagram, there are normal methionine, alanine, cysteine, lysine, adenine, and these are the amino acids. So, whenever at second codon, C is replaced by T, then the amino acid alanine remains the same. So, GCC codes alanine as well as GCT also codes for alanine. So, there is no change in amino acid, so no change in function of protein. And that's why it is called as a silent type of mutation. In the second case, at the third genetic codon, instead of C changes to A, so TGC changes to TGA. TGC codes for cysteine, but here TGA is a termination codon. So therefore there is no further protein synthesis and it is called as a nonsense type of substitution mutation. In the next case, at the third position in the normal, TGC, here instead of T there is a replacement by G. Then the amino acid cysteine is replaced by arginine. So there is a change in amino acid and that change may be responsible for change in function of this protein. Therefore, this type of mutation is called as missense type of mutation. The next type of point mutation is frame shift mutation. So due to insertion or deletion of one or more nucleotides, the reading frame of genetic codon changes like a sentence and therefore the function of protein also changes. For example, in the original, the genetic codes are like this: fat, cat, 8, DB, rat. But if the frame changes or shifts, that is A is added at this position, then the sentence changes to fat, but instead of cat, it changes to CA. So further all the words are changed. So the sentence become fat, cat, tet, you, ear, and. So it is a meaningless. Therefore, due to the frame shift mutation, the abnormal protein is synthesized. Thank you.